Hi, I'm Dr. Christina Blanchard-Horan, founder of GHL Solutions and co-author of this course, Clinical Trials in 7 Easy Steps. I've spent 20 years in the field of clinical trials, working with NIH and CDC on HIV trials in international setting. My job was to prepare clinical trial sites, work with them to ensure they met all regulatory guidelines in preparation for conducting clinical trials for federal studies. So let's get started with lesson 1.
Hi, I'm Jabin Sharma and I'm the co-author of Clinical Trials in 7 Easy Steps. Having worked as a clinical research coordinator in India, I found it extremely difficult to find a job in the same field once I migrated to Canada. Understanding the plight of many other immigrants and newcomers who often have a background in life sciences but are unaware of the opportunities available in clinical research, I decided to come up with a course that would help aspirants understand clinical research trials and the opportunities to work in this field. I have known Christina for over 15 years as we have collaborated together in clinical trials for HIV and AIDS. Christina helped me put my experience and thoughts to reality as we work together on coming up with this clinical trials course. This course will help those who want to pursue a meaningful career in this industry by providing them with an understanding of the different aspects of clinical research trials. I hope that this course will provide many people with the opportunity to pursue a rewarding career in clinical research.
The purpose of lesson 1 is to develop the knowledge and basic understanding of the types of clinical trials there are and the terminology that goes along with it. The goal of this lesson is for students to gain a general understanding of clinical trials to give them the foundation they need to move forward with deeper discussions about clinical trials. And you may ask yourself, why did we decide to talk about clinical trials in the first place? Jabin Sharma, my co-author, and I used Answer the Public to see what people were talking about. This is a diagram of some of the most frequently asked questions about working in clinical trials. Most of them can be found on Reddit, but during the course of the following lessons, we'll be answering the most important questions that you, the public, want to know about. Among others, we'll answer questions like, are clinical trials safe? Why are clinical trials necessary? What's an informed consent? What are potential benefits of taking part in a clinical trial? And what's the difference between a CRA and a CRC role in clinical research? What are some of the things I can do to be working in clinical trials? Those are some of the questions that we'll be answering during the course of this lesson, well, all of these lessons. We've also included the Quick Guide to Clinical Research vocabulary and acronyms, including 20 commonly used terms and acronyms. Some of the resources that you're going to be able to pull from for this course include a book called The Comprehensive Guide to Clinical Research. There'll be a link included below by Dan Sfera and Chris Saab. Now, we highly recommend this book. We're not following it chapter by chapter, but you can find references in the book that you can further explore the depth of clinical trials. And it provides a comprehensive in-depth look at clinical trials itself. Now, we'll use this as a reference book in several of our lessons, but it is not
the full scope of the course. In addition, we'll have a quick vocabulary reference guide, which is also included in the book, but we've included one specific to clinical trials networks. And you'll find a list of acronyms that are commonly used in clinical research and called the Quick Guide to Clinical Research Vocabulary.
What are clinical trials and why are they important today? Clinical trials are research studies that are conducted in volunteer human subjects. Note, I stress that language. Volunteer human subjects, you'll be hearing that a lot. Those are people who participate in clinical trials as subjects, people who are experimented on, basically. You're a volunteer. You must volunteer to participate. And they use these volunteer human subjects to conduct research studies to determine whether new medications or treatments, screening methods, prevention techniques, or diagnostic methods, whether they're safe and effective. So we're looking at safety and efficacy, how safe something is and how effective it is. Now, in order to learn about how a new treatment might work, researchers put patients through sometimes risky series of tests. And these clinical trials or clinical research shows if the medication or the device is safe and effective for use in humans before it goes onto the market to be sold to the general public. In these studies, researchers look at how people respond when they're given whatever the intervention is, whether it's a prevention, a placebo, which we'll talk about later, or a medication. They look at what intervention, that is the intervention, so that scientists can then tell if the medication or the intervention is safe and effective for the needs of the person using the intervention. This means that clinical trials often serve as both research tools as well as potential therapies.
What makes clinical trials important today? The importance of clinical trials today can be captured in the questions that arose around the process for bringing the COVID vaccine to market. Usually, approvals take many months and sometimes years to get. So how was Pfizer and Moderna able to get their vaccines through the approval process so fast? Was it legitimate? Have the vaccines met our safety standards? We will use this Pfizer vaccine as a case study to demonstrate a clinical trial story that will answer these questions. In general, clinical trials research is designed for discovering new treatments and new vaccine preventions, reducing the chance of developing diseases. They can show researchers what does or doesn't work in humans that cannot be learned through experimentation with animals, but only if it's done correctly. Clinical trials test the safety and efficacy of the drug or device and bring new treatments to the market. Most of all, they remove the physician's bias by reducing the chance of developing diseases, showing researchers what does or doesn't work in humans, testing the safety of the drug, vaccine, or medical device like hearing aids or prosthetics, ensuring the efficacy of the drug or device, bringing new treatments to the market.
There are many types of trials, which we will not list here. You'll find that in an insert of a comprehensive list from the book, Dan Svera and Chris Sauber. We're going to focus on the 4 main types of clinical trials. These are prevention, treatment, screening, and quality of life. Or the most commonly used types of studies are prevention and treatment trials. That's where our experience lies. So prevention trials test whether a particular intervention can prevent the development of a disease or condition. And these types of trials are typically conducted with healthy people who have not yet developed a disease or condition that's in question. Treatment trials test whether a new treatment or regimen is more effective, safer, easier to tolerate than the existing standard of care. Now, these trials are conducted with people who've already been diagnosed with the disease or condition in question. There are screening trials which test whether a new screening method is more effective than the existing standard of care. These trials are conducted with people who've not yet been diagnosed with the disease or condition, but who are at high risk for developing the condition. And then there are quality of life trials that test whether a new intervention can improve the quality of life for people who have a chronic disease or condition like diabetes. These trials are conducted with people who have already been diagnosed with the disease or condition in question. Question: what are clinical trials? If you said that clinical trials are research studies, that they are conducted in volunteer human subjects, that they determine safety and effectiveness of medications or treatment or techniques or screening, etc., then you were right. Prevention studies are vital to our understanding of how best to protect people from disease. By testing new treatments and interventions, we can learn how to better prevent or even cure diseases. The purpose of prevention studies is to find ways to prevent people from getting certain diseases or conditions. And this may involve testing new treatments or interventions like vaccines or medications to see if they can help prevent disease. By testing these new treatments and interventions, we learn how to protect and provide better public health. Prevention studies include 4 main types. 1, the primary prevention studies aim to prevent the onset of a disease or condition in people who have never had the condition before. The second type of prevention studies aim to prevent the progression of a disease or condition in people who already have that disease. Tertiary prevention studies aim to prevent the complications of a disease or condition that someone already has. And quaternary prevention studies aim to prevent the reoccurrence of a disease or condition in people who already had the disease or condition. There are many different types of prevention studies, such as clinical trials, observational studies, and epidemiological studies. Clinical trials test new treatments or interventions in people to see if they're effective at preventing the disease. Observational studies look at how people's lifestyle choices, such as diet and exercise, affect their risk of developing a disease. Epidemiological studies help us understand how diseases spread and what factors contribute to the development of disease. The HIV Prevention Trials Network, or HPTN, is a worldwide collaborative clinical trials network that develops and tests the safety and efficacy of interventions designed to prevent the transmission of HIV. The network's main focus is on prevention of HIV disease. The HPTN studies strive to prevent HIV infection through the use of antiretroviral drugs, interventions for substance use, behavioral risk reduction, and structural interventions. That's changes within the structure of systems. HPTN research studies are developed by protocol teams that include clinical trial unit investigators and recognized experts in HIV prevention. As study protocols are developed, they undergo a rigorous intra and extra network review process. Everybody's looking at these to ensure that they're in compliance with current ethical guidelines and regulatory procedures. HPTN 052 was a phase 3, 2-arm, randomized, controlled, multi-center trial. Ooh, look at all those vocabulary words for you to learn. We're going to learn each one of these. Anyway, HPTN 052 phase 3 trial was implemented to determine whether antiretroviral therapy, or ART, can prevent the sexual transmission of HIV-1 in HIV-1 serodiscordant couples. That's couples where one person is HIV positive and the other is not. So, what makes this prevention trial different from other screening studies? Well, treatment research generally involves an intervention like a medication or psychotherapy, a new device, or some new approach to surgery or radiation therapy. Prevention research looks for better ways to prevent disorders from developing or returning. This study was conducted in 13 sites located in 9 countries: Brazil, Botswana, India, Kenya, Malawi, South Africa, Thailand, and the United States, as well as Zimbabwe. To learn more about the HPTN 052 study, you can go and click on the link below. Screening trials or screening studies aim to improve screening for disease. Now, the process, known historically by its terminology as screening, involves all steps performed after acquiring a participant's informed consent, but prior to actively recruiting the study participants into an experiment. This includes doing things like sending questionnaires or just interviewing potential subjects. Regardless of whatever the strategy is used, investigators must protect the privacy of the potential participant and the confidentiality of the information collected about him or her or them. We will be offering this topic in more depth in the next lesson. The idea of screening trials is to evaluate new tests for detecting health conditions in people before they have the symptoms. This way, you can determine whether or not the test saves lives and what cost would be involved with it at any given time. The main objective behind these types of studies is if they were done clinically on volunteers who possess no known disorders that might account for their condition. Researchers won't make sure that there isn't anything wrong other than simply being prone towards developing certain diseases later on down the line. The methods of detecting disease, often called screening tests, can include imaging tests that produce pictures of certain areas of one's body, laboratory tests like a blood test or a urine test, genetic tests that look for inherited genetic markers. A genetic marker is a specific gene or other identifiable portion of DNA that can be used to identify an individual disease or trait. To learn more about this, click on the link below. So here's an example of a screening study. The title of the study is Improved Neuropsychological and Neurological Functioning Across 3 Antiretroviral Regimens in diverse, resource-limited settings. And this was conducted by the AIDS Clinical Trials Group, study number A5199, the International Neurological Study, which I actually assisted with the development of. The background for this, the AIDS Clinical Trials Group, this particular study, A5199, the psychological and neurological functioning across ART, compared the neurological and neuropsychological effects of 3 antiretroviral regimens in patients infected with HIV-1. And they did this in resource-limited settings. And so the participants who participated were in Brazil, India, Malawi, Peru, South Africa, Thailand, and Zimbabwe. And they were randomized to 3 ART treatment arms. And then they were standardized and the screening examinations were done. So this study detected really no significant differences in the outcomes of these 3 regimens. The study suggests that treatment with either the WHO recommended first line or for resource limited settings would actually improve neurological functioning and reduce neurological dysfunction. So this was a very important study. They really looked only at screening and screened patients, and then through that screening were able to draw conclusions. Quality of life studies are different. Clinical trials are an important way to measure how well treatments work and impact people's quality of life. For example, cancer patients might experience side effects such as nausea or vomiting, which can be reduced with new drugs tested through clinical studies. Now, the goal is to develop new drugs that provide the same treatment but with fewer side effects. That's a quality of life study. Clinical research helps us find better ways for better living, despite our illness. The quality of life is an abstract concept with different philosophical and societal and health definitions. Health-related quality of life focuses on the aspects that are related to illness or treatment in some way. The measurement of patient-reported outcomes, including health-related quality of life, also called HRQOL, if you're keeping up with your acronyms, is a new initiative which emerged and grew over the past 4 decades. So the last 40 years we've been doing quality of life studies. So these assessments include more than just one domain, like performance status or symptoms. It can also take into account a patient's reported assessments needs, like satisfaction with their care or a complete picture for how one is doing in the daily lives that they lead. And those kinds of studies can be done to determine how to improve quality of life. The term patient-reported outcomes was introduced in 2004, and it was to include data that were reported directly by the patient and without intermediary or like their family member or a health professional interceding. The process of obtaining patient-reported outcome information is sometimes referred to as PROM, Patient-Reported Outcome Measurement, PROM, another acronym for your feather, for your cap. All right, let's move on. Treatment studies or treatment trials. These studies are different from prevention trials, and what makes a treatment trial not a prevention trial? Well, let me explain. Clinical treatment trials are an essential part of our drug development process, and they play a major role in determining which medicines will be available for public use. So there's often 4 different stages in these types of studies, and each with its own purpose that helps scientists to figure out what questions need to be answered and before moving on towards another phase. So each phase has its own requirements, and you don't move to the next phase until you complete the phase before. And there's one thing that all these studies have in common. They're designed to answer questions about how well therapies work when they are taken as prescribed by the doctors. Now we're going to talk more about treatment studies. Question: what type of trial is this? A trial that enrolls pregnant mothers who are HIV positive to see if they can test a certain drug to prevent HIV transmission to the infants that the pregnant mothers will deliver. What do you think it is? If you said treatment trial, you were right. The phases of clinical trials are preclinical, phase 1, phase 2, 3, and FDA review, and phase 4. Drug interactions or adverse reactions are tracked during all of these phases and beyond. In fact, marketing trials continue after a drug is on the market. So this list of phases does not include marketing trials. Let's look at the phases of the mRNA vaccine that Pfizer and Moderna used for COVID. So here we are, Pfizer's landmark COVID-19 vaccine trial, a phase 1, 2, 3 placebo-controlled, randomized, observed, double-blind, dose-finding study to evaluate the safety and tolerability, immunogenicity, and efficacy of the SARS-CoV-2 RNA vaccine candidates against COVID-19 in healthy individuals. Now, let's look at this. mRNA COVID vaccine. The purpose of the study was to evaluate the safety, tolerability, immunogenicity, and efficacy of SARS-CoV RNA vaccine candidates against COVID-19 in healthy individuals. Now, the purpose of the study was to rapidly describe the safety, tolerability, and immunogenicity of two, and I'll say this, BNT162 RNA-based COVID-19 vaccine candidates. So there were two candidates, and they were both examined against the COVID-19 and the efficacy of one candidate in healthy individuals. So there are currently no licensed vaccines to prevent infection with SARS-CoV-2 or COVID-19, and given the global crisis of COVID-19 and the fast expansion of the disease in the United States and elsewhere, the rapid development of an effective vaccine was of utmost importance. Therefore, let's see how that was handled. The preclinical phase is where they're deciding whether a drug is really ready for clinical trials, and it involves an extensive preclinical study that yields preliminary data about the efficacy, the toxicity, that means how toxic the drug is, and the pharmacokinetic information, that's how long it stays in your system. Wide doses of the candidates are tested using in vitro, so test tube or cell culture and animal experiments. Now, it's also possible to perform computer modeling exercises where scientists actually use algorithms based on protein-protein interaction, networks and predictions, things they've already looked at and tested, and they can compare them and see about how well these drugs will work against specific targets. when given at high doses or versus low doses. Before a drug can be tested in people, it first needs to go through extensive testing in test tubes and animals. Now, this helps scientists learn how well the drug works and how safe it is. The drug is also tested at different doses to see what's the most effective and most safe dose. There was a lot of fuss around the fact that the COVID vaccine came out so quickly. However, a lot of people don't realize that the preclinical trials had already been completed on SARS-2. Preclinical studies showed that 2 doses of the vaccine protected mice against infection of the nasal mucosa and the lungs. So in other words, it prevented or helped to protect them by giving them the adaptive SARS-CoV-2. In phase 1 clinical trials, the first tests of a new medication or treatment are tested on a small number of people. This usually involves about 20 to 80 people, and the goal of those tests is to see if the drug is safe for use in humans, or if it has some bad side effects. If it does, then the trial stops. If it doesn't, then the next phase of testing begins. In phase 2 trials, they're different because they're really figuring out if a drug or a treatment is effective and safe. And this phase usually involves 100 to 300 volunteer participant subjects, and these people help to figure out how the drug or treatment works, including any potential side effects. So this information will help make decisions about whether to continue with phase 3 trials. In phase 3 trials, these are very important. They help researchers learn if a drug is effective and how it might affect people. These trials have between 1,000 and 3,000 participants, and it helps researchers know if the drug is safe and also how it might work in people. So they monitor side effects in larger numbers of people. And this goes on, this monitoring of patients and their side effects, etc. There is a database that tracks this, and it's monitored very carefully. Phase 4 clinical trials. After a drug is approved by the FDA, it is important to study how people are responding to the treatment. So this can help doctors make better decisions about what patients should use or what they should avoid. The post-approval marketing study provides detailed information about side effects and proper dosage instructions. This is where pharmaceutical companies and FDA continue to monitor the side effects of drugs that are produced and also proper dosages, the effects of the drugs. In this course, we look at a number of studies. Some of them include, for example, PEARLS, which was looking at combinations for treating HIV naive people in resource-limited settings. The MOMS study, which explored options for pregnant women who were HIV positive to prevent resistance. And then there was the STRIDE study looking at early ART treatment in people with HIV positive and tuberculosis, to name a few. You can read more about these studies in the information below. There are links to each of the studies where you can explore and learn more about those studies and the resulting literature. Let's move on. Okay, this is the end of this lesson. You may complete the quiz and move on to the next lesson.